presentation, we will explore cell-mediated immunity by focusing on the activation of cytotoxic T cells and the process by which they rid the body of foreign invaders. Now, all nucleated cells in the human body contain a molecule called MHC1. If the cell becomes infected, it uses the MHC1 molecule to display a fragment of the antigen on the surface. This is called an epitope. The cell's saying, "I'm infected, come and help me." Cytotoxic T cells contain a molecule called CD8. This enables the T cell to dock with the MHC1 molecule on the infected cell in a process called co-stimulation, whereby the T cell receptor on the cytotoxic T cell recognizes the epitope on the infected body cell as a match, and it becomes activated. This begins a series of reactions that will ultimately kill the infected cell. Now, we've looked at the process by which a cytotoxic T cell becomes activated, but how does it go about ridding the body of the infected cell? Well, first, the original T cell divides and differentiates into memory T cells and effector T cells. Memory T cells are inactive, but they act as guards, ready and waiting, and if the same antigen enters the body again, bang, they become active and mount an even stronger defense. Effector T cells, however, they're already active. They go straight into battle and destroy other infected cells produced by replication, such as this little one here. So in order to kill the target cells, the cytotoxic T cells release these lytic granules, which are modified lysosomes that contain three effector proteins: perforin, granzymes, and granulysin. This causes apoptosis of the target cell, which is programmed cell death. The exact method by which this happens is still not completely understood, and there remains a number of conflicting ideas. The most widely accepted theory, however, states that perforin forms these little pore-like structures that embed themselves in the plasma membrane of the infected cell. These pores allow extracellular fluid to flow into the cell. This causes osmotic instability within the cell and ultimately results in cytolysis, which is cell bursting. Once granzymes leave the lytic granule, they enter into the target cell by endocytosis, which involves binding to specific membrane receptors such as mannose-6-phosphate receptors. Granzymes themselves cannot directly fragment the DNA within the cell's nucleus, so it's believed they activate these enzymes called CPP32. These are responsible for DNA degradation and ultimately apoptosis of the cell. The third protein, granulysin, this causes apoptosis by entering through the little pores caused by perforin. This disrupts the negatively charged synthetic liposomes, causing holes in the cell membrane. This form of apoptosis is believed to be the most prevalent in tumours. Now, cytotoxic T cells can also kill target cells by releasing specific toxins such as lymphotoxins, but in this presentation we mainly focused on granule-mediated cell death. So once apoptosis is complete and the cell is dead, the cytotoxic T cells move away and the remains of the target cell are ingested by phagocytic cells.